Hello everyone, welcome back. Our lecture sessions, overview and integration of cellular metabolism. We are at the very end of our class. This is the 60th lecture of this NPTEL lecture session, and it is about a very important topic, that is metabolism of cancer cells. These are the concepts we are going to discuss in this class. What is the unique metabolic characteristics in cancer cells? One very important phenomena, Warburg effect. Then what are the different metabolic pathways, or rather the metabolic pathways which are occurring differently from normal cells in cancer cells, we are going to discuss. What are the drivers of these type of metabolic changes, or rather metabolic reprogramming? Then finally, how this metabolic reprogramming can be exploited as a therapeutic target. So, we are going to cover these in our this lecture session. So, let us start. Brief about cancer cells. Actually, what happens in cancer cells, you all know, the normal growth control mechanism is lost in cancer cells. There is uncontrolled cell division, uncontrolled cell proliferation, which is finally leading to formation of tumor mass. Now, metabolism of tumor is not a sole tumor cell metabolism, rather it is influenced by the surrounding cells, surrounding blood vessels, the metabolic environment surrounding the cancer cells, metabolic environment inside the cancer cells. So, that is a holistic metabolic milieu, and that is called tumor microenvironment or TME. So, basically TME is the effect which is imparted over the surrounding stromal and immune cells, extracellular matrix, blood vessels, the effects imparted over these non-cancerous tissues by the cancer cell, as well as how these tissues and their metabolic environment can affect cancer's metabolism. So, that is the tumor microenvironment, and that can be delivered via different metabolites, different signaling molecule, and also proteins in the intracellular space. So, as a whole, this forms the basis of the metabolic reprogramming in cancer cell. Now, what is the metabolic reprogramming? Definitely these are the changes of metabolism inside cancer cells, which is different from the normal cells, and those have been adapted for the survival of the cancer cells, for the therapeutic evasion of the cancer cells, metastasis or spread of the cancers throughout the body, and also how they exploit the surroundings for their own nutrients. So, that is the metabolic reprogramming in cancer cells. Now, remember, all the cells in a cancer are not actually showing same type of metabolism, not even two different cancer cells are showing different types of metabolism. Suppose a cancer in kidney, renal cell carcinoma, a cancer in lung, they might not be similar from the metabolic perspectives. So, it is metabolic heterogeneity we call. So, this can be intratumoral diversity in different subpopulation, as well as tumor to tumor variation definitely exists. Now, why this metabolism in cancer cells should be studied? Because definitely this is the important thing which is finally helping tumors to develop, to progress. So, those valuable insights are needed for understanding how tumor is processed. Identifying the key mechanisms, who is the driving factor for the growth of the cancers, we can understand from it. Then finally, we can develop different therapeutic targets, the metabolism which are specific for tumor or different to the tumor cancer cells, we can target those pathways to develop effective treatment module. So, these are the purpose of studying metabolism in cancer cells. Now, in 1924, Otto Henrik Warburg, he is a very famous Nobel laureate for his groundbreaking work in the field of cancer metabolism, which started very early in 1924. Can you imagine? It is 1924. After around 50 to 100 years, actually, after centuries rather, when the cancer metabolism is presently is gaining the attention in recent days, but in very early time he actually observed a very peculiar phenomena when his colleagues, when the similar researchers are actually on the speculation that tumor cells are gaining energy from uncontrolled replication of proteolysis or lipolysis, from protein and lipid actually. Warburg observed that cancer cells, it uptakes glucose very rapidly and convert them into lactate even in presence of oxygen. Remember, this is very important. Even in presence of oxygen, if you remember, in glucose metabolism, we have talked about glycolysis as well as TCA cycle. Now, if you remember once again, glycolysis occurs in aerobic condition as well as anaerobic condition. The anaerobic fate of glycolysis is formation of lactate from pyruvate, whereas in case of aerobic glycolysis, pyruvate enters the TCA cycle, and in absence of oxygen, anaerobic glycolysis is preferred. But in cancer cell, even if there is oxygen supply, cancer cell prefers glycolysis. So, it converts pyruvate to lactate and shows a very rapid consumption of glucose, and this phenomena is coined as Warburg effect. It was long ignored, rather long not studied when it has been postulated by Warburg, but later it gained the attention and the term was and the phenomena was coined Warburg effect, and this is the groundbreaking discovery which gained the attention towards cancer metabolism. Now, what is the difference between the normal cellular proliferation versus tumor cellular proliferation? Remember, tumor cells are actually they have lost the controlled cellular proliferation. What is controlled cellular proliferation? Which occurs in mammalian cellular mammalian cells, non-cancerous cells. Basically, cellular proliferations are based on some extracellular stimuli like growth factor. One very important stimulus is growth factor. Now, these growth factors interact with the receptors, activate some intracellular signaling cascades, and it delivers a very finely controlled mechanism for the cell division, and that also depends on, it is not just the cell's property, it is also depending on the neighboring cells, different chemical signals, its microenvironment and also the cell itself. But in case of cancer cells, what happens, these growth receptors, there is some mutation which causes increase in growth factor receptors. So, there are increased receptors, also the intracellular signalings which promote the cellular proliferation, they are constantly switched on or amplified. So, there are increased receptor, those receptors are giving some amplified signal, and tumor cells are dividing rapidly. Now, for rapid cell division as well as this enormous tumor mass, they need more nutrient, they need more biomolecules. For that, biosynthetic capability of the tumor cells should be increased, like fatty acid supply, nucleotide supply, those should be increased in amount, and majority of the tumor's metabolism is basically targeted or fulfill this target, that the tumor needs more biosynthetic molecules for its uninterrupted proliferation. And finally, metabolic and autonomy. As I told that in the normal non-cancerous cells, the cell division is based on the surroundings. Here it is not. The cancer cell gains autonomy for its proliferation. So actually through metabolism, what it needs? More energy, more molecules for its proliferation, and that is the ultimate target of cancer cell metabolic reprogramming. Now, this Warburg effect, which is also known as aerobic glycolysis, what are the advantages of this? Why this aerobic glycolysis is there? Number one is, instead of going through glycolysis and TCA cycle, which actually supply from one molecule of glucose, it actually supplies more ATP in comparison to glycolysis or formation of lactate from pyruvate via glycolysis. From one molecule of glucose, there is rather less number of ATP, but remember, only glycolysis is of short duration. So, what is done in cancer cell? There is multiple cycles of glycolysis in a short duration, which actually generate more ATP per unit time. So, there is rapid glycolysis, rapid ATP production, and these glycolytic intermediates are actually the source of carbons required for different biosynthetic pathways. What are those? Glucose 6-phosphate, if you remember, it enters the pentose phosphate pathway to generate two important molecules. One is NADPH, another is molecules for nucleotide synthesis. Ribose 5-phosphate is actually provided from pentose phosphate pathway. And another one very important biosynthetic pathway is shunting the 3-phosphoglycerate into serine and glycine biosynthetic pathway. How? You can see in this cell, 3-phosphoglycerate can form 3-PHP, which is 3-phosphohydroxypyruvate. With the help of the enzyme 3-phosphoglycerate dehydrogenase, where NAD is utilized, and PHP is converted to 3PS, phosphoserine, with the help of the enzyme PSAT, which is phosphoserine aminotransferase, and finally the phosphoserine is converted to serine with phosphoserine phosphatase. So, finally, you can see serine can be synthesized from 3-phosphoglycerate, and from serine there are multiple biosynthetic pathways can be followed, like formation of phosphatidylserine, which helps in membrane lipid formation, sphingolipid formation. Then serine can be converted to glycine via serine hydroxymethyl transferase, and then glycine is helpful for formation of glutathione, and then here you can see methionine also is taking part in this pathway. So, basically these amino acids are important, as well as biosynthetic pathway can be followed from the glycolytic intermediates. Then NADPH, you know, NADPH is important for reductive biosynthesis of different types of lipid, membrane lipid, then fatty acid synthesis. Phospholipid biosynthesis is enabled by conversion of dihydroxyacetone phosphate to glycerol 3-phosphate. Also fructose 6-phosphate, one intermediate of glycolysis, can enter the hexosamine pathway, and this pathway is important for different post-translational modifications of proteins, which are important in the survival of cancer cells. And ultimately we have discussed about glutathione, how it can be, how glutathione formation can be helped via glycolysis. Now, glutathione is very important for cancer cell survival, because remember, when there is TCA cycle, the products while going through the electron transport chain inside mitochondria, those are the sites where free radicals are formed, and much flux through the TCA cycle and electron transport chain, there is much formation of ROS or reactive oxygen species. Now, just assume that when there is high rates of proliferation in cancer cells, if it would have been the condition that much high metabolism is going through metabolic flux is going through glycolysis, TCA cycle and electron transport chain, there would have been high production of ROS, which would have been harmful to the survival of cancer cells. Even whatever ROS is produced in the cancer cells can be dealt via increased glutathione production. So, these are the helpful effect of Warburg effect or aerobic glycolysis over cancer cell survival. Now, in aerobic glycolysis, what are the enzymes or what are the proteins which are altered in their expression, starting from the very glucose transporters which are overexpressed, GLUT1, GLUT3, these are the glucose transporters very much expressed over the cancer cells, and those actually cause rapid uptake of glucose inside cancer cells. Different glycolytic enzymes are overexpressed like hexokinases, these are the isoforms I have mentioned, triose phosphate isomerase, phosphoglycerate kinase, enolase, pyruvate kinase M2, aldolase A, phosphoglucomutase, pyruvate dehydrogenase, these are a lot of enzymes rather in the glycolytic pathway is actually overexpressed. Moreover, the very important regulatory bifunctional enzyme phosphofructokinase and fructose 2,6 bisphosphatase. Remember, from the class of glycolysis and neoglucogenesis, we have discussed how they are regulated. We have discussed this enzyme, which is a bifunctional enzyme, phosphofructokinase, fructose 2,6 bisphosphatase. This is also activated or rather differently expressed in comparison to the normal cells in cancer cells. How? You can see that this is the GLUT1 transporter which is overexpressed in cancer cells. So, there is increased glucose uptake which is converted to glucose 6-phosphate via increased expression of hexokinase. Now, hexokinase forms fructose 6-phosphate, and here the enzyme phosphofructokinase, which is a bifunctional enzyme, is important. Now, you can see there are different isoforms of phosphofructokinase. Two such isoforms are phosphofructokinase fructose bisphosphatase isoform 3 and phosphofructokinase fructose bisphosphatase isoform 4. Now, these two are actually acting differently. Phosphofructokinase fructose bisphosphatase 3 isoenzyme is actually helping in formation of fructose 2,6 bisphosphate. So, basically what it is causing, it is increasing the flux through glycolysis. Remember, if there is more formation of fructose 2,6 bisphosphate, which is one allosteric activator for the glycolysis. So, if there is more formation of fructose 2,6 bisphosphate, what it causes? It increases the flux through glycolysis. But to its contrary, phosphofructokinase fructose bisphosphatase 4 isoform, it basically converts the fructose 2,6 bisphosphate to fructose 6-phosphate. So, basically these phosphofructokinase fructose bisphosphate isoform 4, it inhibits the flux through glycolysis, causes accumulation of fructose 6-phosphate, which in backflow causes accumulation of glucose 6-phosphate. So, glucose 6-phosphate can be diverted towards pentose phosphate pathway. Now, there is another enzyme which is also known as TIGR. TIGR stands for TP53 induced glycolysis regulatory phosphatase. Now, this enzyme along with phosphofructokinase, fructose 2,6-bisphosphatase, isoform 4, they together inhibits the flux of glycolysis, rather favors the flux of pentose phosphate pathway. Now, it has been seen that both of these isoform are actually expressed in different tumor cells based on their requirement. So, even pentose phosphate pathway is increased, which is helping the cancer cell via formation of nucleotide and NADPH, which are actually delivering multiple roles in the tumor cell metabolism, as well as glycolysis. If glycolysis is favored, there will be formation of pyruvate, which will finally form lactate. Now, very important, lactate formation is also required for the cancer cell survival. Why? We are going to discuss. You can see that in cancer cell, or rather in cells actually, lactate is secreted out from the cell via one transporter, monocarboxylate transporter, monocarboxylate isoform 4, and their expression is also increased in tumor cells, causing more release of lactate from cell in to the cellular surrounding environment. So, what are the drivers for this Warburg effect? One very important pathway, or rather one very important kinase, enzyme activation, plays an important role in delivering the aerobic glycolysis. That is one serine threonine kinase, which is also known as protein kinase B, AKT. Their activation causes as a whole increase of ATP level through the regulation of both glycolysis as well as oxidative metabolism, which finally, what is the effect? There is increased oxygen consumption. Similarly, glucose transporters are more expressed over the cell surface. So, basically they are translocated more towards the cell surface. Hexokinase expression is increased, and also in glycogen metabolism, glycogen synthase kinase is also inhibited here. Then this pathway, activation of phosphatidylinositol 3 kinase PI3K AKT pathway, activation of this pathway makes the cell more glucose dependent, so that the energy mostly comes from the glucose, because in cancer cell what happens, beta oxidation of fatty acid is actually inhibited. So, Warburg effect is not only helping to supply the biosynthetic molecules to the cells, it also helps in tumor cell survivals via evading apoptosis, as well as its spread via inducing metastasis. How? Now, there is one very special type of apoptosis which is known as anoikis, where if the cells are detached from the extracellular matrix, the accumulation of reactive oxygen species, they triggered this anoikis. Now, anoikis can be exempted in case of cancer cells. How? Because in cancer cells, as I told you, mitochondrial ROS formation, reactive oxygen species formation is reduced, not only via synthesis of increased glutathione from increased NADPH production, but also there is low flux through oxidative phosphorylation, which is a soft target for ROS production. So, basically ROS production is decreased, which actually triggers the anoikis. So, this extracellular matrix detachment in cancer cells, this phenomena can be uninhibited, causing not only evasion from metastasis, but also helpful, not only evasion from apoptosis, but also helpful in metastatic dissemination of tumor cells, that is spread to distant sites within the body. Apart from that, there are few therapeutic and diagnostic role of Warburg effect. What is that? In diagnosis, PET is positron emission tomography. Positron emission tomography is one imaging technique where radio labeled glucose analog, which is fluorodeoxyglucose, is utilized to detect those areas where high glucose consumption is there, and this enables the detection of cancerous tissue. So, here this is how increased glycolysis or increased consumption of glucose is exploited in diagnosis of the tumor cells, or rather specifically locating where the tumor cells are. And finally, outcome prediction can also be helped, because these glycolytic patterns or rather increased glycolysis is actually a poor outcome, and that can be studied via different gene expression profiling. Then metabolism in cancer cells and their microenvironment can be different. As I told you, there is heterogeneity in this microenvironment. Now, cancer cells can be hypoxic, can be normoxic, as well as there is acidosis which is present in cancer cell, because I told you there is huge amount of lactic acid production which causes lactic acidosis or a generalized acidosis surrounding the tumor cell, because lactate is actually secreted. And it is also seen that partial pressure of oxygen in tumor cell is frequently lower than the surrounding normal tissue. Now, this intratumoral hypoxia by prognosis, it has larger risk of local spread, distant spread in terms of metastasis, and also poor therapeutic outcome and mortality. Also, hypoxia renders resistance of the tumor cells towards mitochondria dependent apoptosis. Also, the metabolic reprogramming of tumor cells, which changes the metabolic flux through the mitochondria, Krebs cycle, etc., they are actually the effect or rather adaptation of the tumor cells toward these hypoxic conditions. And for that, one very important molecule should be named, that is hypoxia inducible factor, HIF1. Now, this hypoxia inducible factor, its activation is related to different gene expression, genes or rather proteins or enzymes of glycolysis, enzymes or enzymes of lactate production, different transporters of lactate or proton transporters. Not only that, tumors own phenomena like angiogenesis or formation of new blood vessels, then metastasis, also iron metabolism, those are influenced by hypoxia inducible factor. Now, hypoxia inducible factor enhances different glycolytic enzymes, expression of different glycolytic enzymes, glucose transporters like GLUT1, 3. What are the lactate production related enzymes? Lactate dehydrogenase, told you the transporter, monocarboxylate transporter, their expressions are enhanced by HIF. Also, in pyruvate dehydrogenase kinase expression is induced, then pyruvate dehydrogenase activity is inactivated. So, basically entry to the TCA cycle is inactivated, and finally, fate of cancer, severity of cancer can be predicted by the expression of HIF1. Hypoxia inducible factor, as you can see, it is basically one heterodimeric complex. There are two subunit, HIF1 alpha and HIF1 beta. Now, the beta form is constitutively expressed, constantly expressed, not influenced by others. that is constitutive expression of a gene. So, HIF1 beta, its expression is not changed, it is constitutively expressed, whereas HIF1 alpha is basically dependent on the oxygen availability. So, how it is influenced? By the presence of oxygen? Oxygen causes, the presence of oxygen causes its degradation, that is why it is known as hypoxia inducible factor. Fine. Now, what happens in presence of oxygen? As you can see, in presence of oxygen, HIF1 is hydroxylated with the enzyme prolyl hydroxylase. Prolyl hydroxylase domain protein is also known, it is also known by that. So, the enzyme is prolyl hydroxylase, and this prolyl hydroxylation is recognized by another protein which is known as VHL, von Hippel-Lindau protein, and this von Hippel-Lindau bound HIF1 alpha is targeted by proteasomal degradation. So, these are the phenomena which happens in presence of oxygen, and this prolyl hydroxylase enzyme is actually activated via the metabolism which is occurring in TCA cycle. But remember, when alpha ketoglutarate is converted to succinate, or there is formation of fumarate as well in TCA cycle, their excess accumulation actually inhibits this proteasomal degradation. Now, what happens in tumor cell? Even in normoxic condition, VHL can be inactive. So, basically proteasomal degradation targeting can be inactive. PhD can be inactivated by accumulation of succinate or fumarate, because remember, the enzymes of TCA cycles are actually their expression is reduced. So, finally, what happens? As I told you, there is accumulation of succinate and fumarate which is finally causing inhibition of PhD. So, it actually generating a situation of pseudo hypoxia, finally inhibiting the proteasomal degradation of HIF1 alpha. Moreover, the AKT pathway, it also increases the HIF's function via its increased HIF1 alpha mRNA translation. So, all these phenomena actually causes increased functional efficiency or synthesis of hypoxia inducible factor, which causes increased expression of different types of glycolytic enzymes or transporters. Now, as I told you, these metabolic reprogramming can be exploited therapeutically. How? There are different therapeutic molecules which are targeted towards this aerobic glycolysis, like glucose transporter, GLUT1 transporter, silibinin. Silibinin is one GLUT1 transporter inhibitor. These drugs causes decreased uptake of glucose in tumor cell. Lonidamine causes inhibition of the enzyme hexokinase. Similarly, glucose analogues like 2-deoxyglucose is utilized, which inhibits the enzyme glucose 6-phosphate isomerase. There are different molecules like in this glycolytic pathway which are inhibits in cancer targeted therapy. Also, enzyme like lactate dehydrogenase A can be inhibited by gossypol, galloflavin. Then glycogen synthase kinase 3 beta can also be inhibited. So, these are the different therapeutic target which is targeted towards this aerobic glycolysis. As I told you, there are different TCA cycle enzymes which their expressions are altered, namely succinate dehydrogenase, fumarate hydratase, isocitrate dehydrogenase. Their effect are mostly over ROS expression. Then cellular metabolism dysregulation via accumulation, it causes accumulation of succinate dehydrogenase which stabilizes HIF. Similarly, fumarate Hydrated is also causing fumarate accumulation, stabilizing HIF. Moreover, fumarate accumulation inhibits enzymes involved in DNA and histone methylation, causing epigenetic changes which favors cancer pathogenesis. Then isosited dehydrogenase mutation, it renders neomorphic activity. Now, neomorphic activities are the newer activity which is rendered in a cancer cell. So, basically what happens, alpha ketoglutarate, instead of proceeding to the normal TCA cycle, forms a new metabolite which is a oncometabolite. Oncometabolites are those whose accumulation is reflected by the toxic effect of cancer cells. So, the new oncometabolite 2-hydroxyglutarate is accumulated from alpha ketoglutarate, and this 2-hydroxyglutarate it inhibits alpha ketoglutarate dependent dioxygenases like histone demethylase, DNA demethylases. So, basically they are also causing defective DNA and histone demethylation, causing epigenetic expression, altered epigenetic expression. So, these are the effect of TCA cycle enzyme mutation in cancer cell. Now, there is a metabolic symbiosis between aerobic and cancer cell. As I told, there is metabolic heterogeneity in cancer cells. In a tumor, few cells are living in normoxic condition, few cells are living in hypoxic condition. Those cells which are actually located far away from the blood vessels, they are actually towards them oxygen supply is low. So, they are in hypoxic condition, but those cells, those cancer cells which are beside those blood vessels, they are in normoxic condition. Now, these normoxic and hypoxic cells, they are in a metabolic symbiosis state. How? So, you can see there is real formation of lactate in hypoxic cancer cells. Now, accumulation of this excess lactate can be harmful for the hypoxic cancer cells. So, what happens? These hypoxic cancer cells, they secrete lactate via MCT4, monocarboxylate transporter 4, and this lactate is taken up by the normoxic cancer cells by MCT1, monocarboxylate transporter 1. And what is the fate of this lactate? Lactate enters the OX-phos system via pyruvate formation through lactate dehydrogenase, and it is getting utilized via formation of ATP. So, you can see there is a metabolic symbiosis in hypoxic and normoxic cancer cells. Then also acidosis. Apart from hypoxia, acidosis is also favoring the tumor cells metabolism. How? Now, why there is acidosis? Definitely there is increased amount of lactate formation. Also, carbon dioxide is a significant source of acidic extracellular pH. So, in the tumor microenvironment, extracellular pH is also acidic. Now, this acidosis promotes extracellular matrix degradation, which is helpful for the invasiveness or metastasis of cancer, local spread of cancer. Also, in normoxic acidosis, here tumor cell inhibits the proteasomal degradation of HIF1, because this nucleolar sequestration of von Hippel-Lindau and HIF1 complex, these complexes sequestration is inhibited by acidosis. Moreover, in the intracellular compartment, there are changes in different other proteins like sodium hydrogen exchanger, their expression changed, monocarboxylate transporter, different proton pumps, also carbonic anhydrases. These their expressions are altered in tumor cells, which renders acidosis not only inside the cell, but also in the extracellular environment. Now coming to one very important metabolism of cancer, that is glutamine metabolism. Glutamine, remember, it is, if you remember that glutamine is the non-essential amino acid, and this is the most abundant amino acid circulating in the bloodstream, and it can be utilized as a source of different biomaterials in tumors. Now what happens, glutamine, the circulating glutamine, it is uptaken inside the cell via different transporters like ASCT2, alanine serine cysteine transporter 2, amino acid transporter, also system N transporter SN2. So these are the transporter which uptake glutamine inside cell. Once inside the cell, glutamine is converted to glutamate via the enzyme glutaminase. Now remember, this glutaminase is the cytosolic glutaminase, whereas in mitochondria there is another glutaminase which is very much activated by the high inorganic phosphate concentration. So finally, even in inside mitochondria, glutamine can be converted to glutamate. So there is increased uptake of glutamine and its hydrolysis to glutamate and ammonia, and this process is known as glutaminolysis. Now glutaminolysis is important in cancer cell. Why? Because remember, glutamate here in cancer cell is important for anaplerosis. If you remember, anaplerosis is basically replenishment of the intermediate of TCA cycle, and here glutamine via formation of glutamate actually replenishing the depleted oxaloacetate in TCA cycle. Why? So once again you can see glutamate can form alpha ketoglutarate via aminotransferase, and this alpha ketoglutarate can be utilized for replenishment of the intermediates in TCA cycle, which causes restoration of the reduced glutathione pool as well, which is helpful for fighting ROS. Also this glutamine is helpful in nitrogen donation for synthesis of purine and pyrimidine nucleotides. Different amino acids can be formed from this the amino group of the glutamine. So glutamine is basically helpful for biosynthetic capability of the tumor cell. Now you can see glutamate can be catalyzed by 3 different amino transferases. You remember, if you remember SGOT and SGPT, here you can see GPT is the glutamic pyruvate transaminase. So pyruvate can be formed, pyruvate can be converted to alanine while glutamate is forming alpha ketoglutarate. Similarly, formation of alpha ketoglutarate can be helped by glutamic oxaloacetate transaminase, where oxaloacetate is converted to aspartate and another amino transferase which is known as phosphoserine amino transferase. So here you can see 3-phosphohydroxypyruvate is converted to phosphoserine by phosphoserine amino transferases, where we are getting alpha ketoglutarate which is utilized in anaplerosis in TCA cycle. Also the other metabolites like serine, as I told, it enters formation of glycine and glutathione are there. So these are for cellular proliferation and they are these enzymes are basically targeted to treat cancers. Similarly, tryptophan in tryptophan metabolism, enzymes like indolamine 2,3 dioxygenase or tryptophan 2,3 dioxygenase, their overexpression actually helpful for the immune targeting of the cancer cells. Basically these cancer cells are targeted by different immune cells, and these enzymes provides immunity from this immunological destruction of the cancer cells. So, basically glutaminolysis rather is helping in biosynthetic properties, anaplerosis of the TCA cycle, and also immunological immunity for the cancer cells. Glutamine metabolism is also helpful for NADPH formation. So, here you can see that the flux of glutamate to alpha ketoglutarate forms malate, and this malate can form pyruvate via formation of NADPH, and this NADPH can be utilized for the reductive biosynthesis as well as ROS scavenging action. How this glutaminolysis is regulated? One very important mechanism is via the oncogene MYC, and this MYC oncogenes product is one important transcription factor which is known as c-MYC. And what is the function of this c-MYC? C-MYC actually regulate the expression of different genes and microRNA related to cell cycle, glucose metabolism, glutamine metabolism, as well as biogenesis of mitochondria, biogenesis of ribosome. So, basically these are exaggerated in a cancer cell through the c-MYC. Now, c-MYC increases the glutamine flux inside the cell via the expression of microRNA 23AB. Basically their transcriptional expression is decreased, which causes increased mitochondrial glutaminase expression, which finally leads to glutamine metabolism. Also the transporter of glutamine uptake, their expression is also enhanced. Moreover, c-MYC also induces the expression of lactate dehydrogenase enzyme, which is helping in cancer cells metabolism. And these are the current therapeutic strategies targeting towards glutaminolysis. So, you can see glutamine depletion can be enhanced, its metabolism can be inhibited via the enzyme inhibition, transporters can be inhibited, different glutaminomimetics can be glutaminomimetics here when it is actually targeted towards the transporter enters the cell, basically inactivates the glutamate dehydrogenase enzyme. Aminotransferases can be inhibited. So, these are the different targets towards glutaminolysis. Then we are coming to the lipid metabolism, which is also altered. The de novo fatty acid synthesis is altered. Now, there are three important enzymes in de novo fatty acid synthesis. If you remember, there is citrate lyase, which is ATP dependent, causes acetyl coenzyme and oxaloacetate formation from citrate, acetyl coenzyme carboxylase, which helps in formation of malonyl coenzyme, and finally fatty acid synthase, which is the main enzyme for fatty acid synthesis and which is NADPH dependent as well. Now, fatty acid synthase is overexpressed, also ATP dependent citrate lyase is overexpressed, acetyl coenzyme also is overexpressed. Moreover, citrate lyase reinforces the Warburg effect by preventing the accumulation of citrate. Cytosolic citrate accumulation is actually diverted towards fatty acid synthesis, and this citrate acts as a glycolysis inhibitor. So, basically aerobic glycolysis can go uninterruptedly. So, it has been thoroughly discussed that glycolysis or aerobic glycolysis is one very important pathway which is enhanced in cancer cell. Rather, cancer cells are depending on this aerobic glycolysis not only for the ATP, but also for different intermediate which is targeted towards biosynthesis. But remember, what happens to the electron transport chain? Definitely it is there in the normoxyl cells. As I told you, there is electron transport chain and ATP production via this pathway, but cells are metabolically flexible. Tumor cells, when there is oxygen available, there is electron transport chain, but when there is depleted oxygen, cancer cells undergo adaptation. You can see electron transport chain in nutrient and oxygen poor environment, they can survive in as low as 0.5% oxygen availability, and their micro environment are adapted via decreasing the ATP demand. Like if you remember, sodium potassium ATPase, one very important transporter which is dependent on the supply of ATP, their requirement is very much reduced, their expression is very much reduced. Moreover, one enzyme kinase, AMPK kinase, which we have discussed thoroughly in obesity, how this AMPK is activated while there is ATP depletion and AMP accumulation, rather AMP is formed from ADP. AMP kinase is activated and that causes adaptation towards the nutrient poor or oxygen poor environment. But otherwise, pyruvate can be derived from glycolysis. Also this fatty acids and amino acids, they can serve the intermediates of TCA cycle via anaplerosis. Also glutaminolysis is causing anaplerosis. Different branch chain amino acids like isoleucine, valine, leucine, they can also form acetyl coenzyme and other intermediates of TCA cycle. So, basically the flux towards TCA cycle is increased, causing increased ATP formation even if there is mutation of the TCA cycle enzyme. If the substrate flux are increased, it might increase the formation of products and finally ATP formation through ETC. So, these are all the metabolic alteration in cancer cells. So, you can see in a normal cell which is very much dependent on the oxygen availability, causing aerobic glycolysis as well as OXPHOS. In cancer cell, there is basically anaerobic glycolysis, which is termed as aerobic glycolysis, which occurs in even in presence of oxygen and forms lactate. Also glutaminolysis, which is very important in cancer cell, fatty acid biosynthesis, which is enhanced in cancer cell, and ROS formed in electron transport chain. Normal cells are vulnerable, but cancer cells are resistant towards formation of reactive oxygen species. So, these are the key points from this cancer cell metabolism, that cancer cell exhibits aerobic glycolysis, which is also known as Warburg effect, from where they preferentially utilizes glucose even if there is oxygen available. Then cancer cell rewire their metabolic pathways and utilize nutrients differently compared to normal cells. They adapt their metabolism to produce more energy or synthesize more biomolecules for their rapid growth and survival. Pentose phosphate pathway is enhanced. Glutaminolysis is one very important pathway which helps in anaplerosis. Fatty acid synthesis, lipogenesis, they are upregulated. Then redox balance, ROS formation is modulated to cope up the cell survival. It also manipulates the tumor microenvironment, its own environment as well as the surrounding environment for its proper growth and evade apoptosis and immune response. And who are responsible for that? Different oncogenes, different tumor suppressor genes, hypoxia inducible factor, they help in metabolic reprogramming of cancer cell. And finally, how this cancer cell can be cancer metabolism can be utilized via designing different therapeutic target. So, here we have discussed all these things. So, we have reached the very last session of this NPTEL lecture series, and it was wonderful taking the totally integrated metabolism as well as highlighting different clinical condition or metabolic diversions from the normal scenario. We have vividly discussed obesity, diabetes mellitus, protein energy malnutrition, as well as cancer metabolism, and as a whole one holistic integration of all the metabolism which is occurring in mammalian cell as well as human body. So, thank you all. See you in the live session. Thank you.